Hello, is my voice audible? Can someone say yes?
Yes, ma'am.
Okay, we'll start in 2 minutes. Someone has raised a hand. Do you want to say something? Gautam Reddy. No? Okay, we will start the session then. So, once I start presenting, I will go into the presentation mode. So, I won't be able to see your comments in the chat box. So I request everyone to turn on their mic and speak if you want to say anything. And we'll be doing the assignment questions this time. So I want you all to answer the questions first because it's already been 1 week, I guess. This is for week 5. So first, you all must answer the question and then I will give you the right answer. Okay? Okay, I hope my slides are visible. Can someone say yes or no if it's visible or not? Yeah, okay. So, welcome all to the week 5 live session of Cell Culture Technologies from NPTEL. The course instructor is Professor Manak Das from IIT Kanpur, and I am Sumati Raviraj from University of Hyderabad. If you have any queries, you can mail me personally to my mail ID, or you can also um, put your queries on the NPTEL portal. Okay, so let's start. So in the week 5, you would have studied regarding the introduction to defined system. What is the defined system? And then mechanical dissociation of hippocampal tissue. So mostly would have studied about hippocampal and neuronal tissue this week. And rules for a mechanical dissociation of the tissue, and drug molecule testing, and adult hippocampal neuron dissociation. Okay, so this is what you have been covered in the past week. So then you have an assignment that you have to submit. So usually I make questions which are similar to your assignment questions to help you solve those, but because this is more theoretical and maybe a difficult assignment, so I have directly taken your assignment questions only. So kindly answer the questions and we can keep it open for discussion. So first question: what is the characteristic shape of a hippocampal neuron? A round, B pyramidal, C elongated, and D hexagonal. Yeah, someone, if you know the
Pyramid.
answer. Yes, yes, very good. So the answer for this is pyramidal. Okay, that is B, option B. So what is this pyramidal shape they're talking about? First of all, what is hippocampus? Hippocampus is a small brain. It is also generally called as a small brain, and it is the structure present near your ears, and it helps you with learning and memory. This is the main function of the hippocampus. The pyramidal shape of the neuron in hippocampus is thought to maximize the efficiency of signal processing and transmission within the brain. So it is the every neuron has, a particular shape. Few neurons are smaller in size, few neurons are very long. You can Google what is the length of the largest neuron in human. So every neuron has shapes and sizes varying according to its function. So the pyramidal shape is to maximize the efficiency of the signal processing and transmission. Okay, and the wide reaching dendrites. So if you know the shape of the neuron, it has a cell body, the axon, the dendrites. So the dendrites, like the processes from the cell body, allow the neuron to integrate a large amount of information, while the long axon allows it to send signals over long distances to the neurons. So like I said, there are the largest neuron in the cell. So it is the axon will be the one that is elongating and covering that long distance. So that will help you to send signals to other tissues or other organs. While the dendrites, the processes from your cell body, is useful to integrate large number of information. So that is why they are also branched and in that particular shape. So this is how it looks. This is your different parts. You can see you have your fusiform gyrus, you have your collateral sulcus, you have your temporal horn of the lateral ventricle, tail of the nucleus, the hippocampal sulcus. So this is how it looks. So you can see the shape and the features of hippocampus. And these are different other uh, smaller parts or regions of it. Okay, so within this organ or this part of your brain, the pyramidal neurons are present. So this is how your pyramidal neuron looks. This is your immunofluorescent staining of neurons from hippocampus. So you can see it has so many dendrites that are arising, and they are connected to different axons and other dendrites and other neurons, and it will conduct uh, electrical impulses, signal transmission. Okay, so these branching of the dendrites and this pyramidal shape will help in its efficiency. Okay, because it is the main center for learning. So when you learn, you need to have storage of whatever you learn. That is nothing but your memory. So for this efficient and increased storage, you need this kind of neurons. Okay, so I hope this is clear. So the next question is, papain is a dash that is derived from dash. A, it is a proteolytic enzyme derived from papaya. B, it is a proteolytic enzyme derived from milk. C, it is a lactose dehydrogenase enzyme from papaya. Or D, it is a lactose dehydrogenase enzyme from milk. So what is the answer for this?
A.
A, yeah, very good. So papain is a proteolytic enzyme from papaya, option A. So proteolytic enzyme means it will help in lysis of your proteins, proteolytic. So most of the scientific names or these kind of classification sort of names, you can understand within the name itself. You can divide the name and think what it could be. So proteo means your proteins, lytic means lysis. So your papain is a protein that helps in breaking peptide bonds of your proteins. Okay, and it is a natural enzyme derived from your fruit papaya. So that is why sometimes few people are suggested to eat raw papaya. Few people are suggested to eat ripe papaya. So the ripe papaya will not have as much as papain, but your raw papaya will have a lot of papain. So for particular dose, a papain is useful even in your body, in your system. But more than that dose, you should not overeat raw papaya. So that high amount of papain is also harmful to your body tissues. Okay, how papain works. Papain cleaves the peptide bonds in proteins, specifically targeting amino acids with large side chains. So your basic amino acid structure, you have an R group, you have a CO group, COH group, and you have a amino group. So your R group or your side chain that your amino acid has, that is where your papain will act. And it is useful in tissue dissociation. Papain digests the extracellular matrix, that is the extracellular component of your cell. So your cell, when it is grown in vitro or in vivo, it will always have an extracellular matrix. Your body definitely has it even when you grow your cells in the culture dishes. Once they start adapting and growing in size and dividing and proliferating, they themselves secrete certain proteins which contribute to the extracellular matrix. Okay, so that is nothing but a glue that holds your cells together. So your cells not only need to attach to a surface if they are epithelial in nature or even endothelial cells, they epithelial cells needs a basement, whereas all the other cells, tissue oriented cells, also need to attach to other cells. So this matrix proteins will help in attaching these cells to other cells. So your matrix proteins will have your collagen, your integrin will be on the cell, your laminins. So all these things will help in adherence, okay, attachment, allowing the cells to separate from one another without damaging. So this extracellular matrix will now be broken by your papain, which will allow your cells to separate from each other but at the same times it will not cause damage to the cell itself. It will only cleave those connections between your one cell and the other cell. Okay. So papain is also used to cleave antibodies. If you have seen experiments with antibodies in the very initial stages, they use different enzymes like papain, trypsin, uh, pepsin, all these things to cleave different portions of your antibody to get different fragments. That is how you know that a antibody has regions like Fab regions, that is your fragment antibody binding region, fragment antigen binding region, sorry, and your FC region, that is your uh, this longer stem of your antibody. Okay, so where your papain cleaves your antibody is in this long chain that attaches your Fab regions and your FC fragment. Okay, so when you treat your antibodies with papain, you will get these kinds of separated regions of your antibody, wherein this is your Fab fragment, this is your FC fragment. There are certain proteins which cleave only here, so you will get Fab 2, like both this Fab will be attached with each other but dissociated with FC, that is a different protein. And then few will, cleave individual uh, individual homomers or heteromers of this antibody. So your papain will act here, cleaving both your this Fab region, and your FC region. So you will get three parts when you cleave your antibody with papain. That is two Fab fragments which are identical, and then your FC fragment. Okay. In cell culture or in tissues, you also use papain to cleave your tissues or your while isolating them, etc., without damaging the cells. So your third question, in hemocytometer, volume of one millimeter into 1 millimeter square is how much? A 0.25 nanoliters, B 5 nanoliters, C 60 nanoliters, and D 100 nanoliters. What is the answer for this? Anybody? Yes, someone please answer. Are you guys there? Yes. At least tell me if you know the answer, if you don't know the answer. Do you know what a hemocytometer is? Have you seen it? You can tell me anything. You don't have to just give the answer. Anything that you know about it, you can tell me. No?
I guess D.
Yeah, yeah. Tell me.
I guess D. 100 ml.
D. D for dog?
Yes, ma'am.
Yeah. So, correct. See, you have to answer. Then only you will know whether you know it or not. It gives you confidence also. So, your answer for this is 100 nanoliters. So, there are different sizes again in the squares based on the size. You have different volume that it can hold. So, when a hemocytometer is a thick slide where you have this very microscopic grading, wherein you will give, see, this is how your grading looks, and here you will give your cells in suspension along with trypan blue or any other dye if you want to stain different type of blood cells. Generally in cell counting for lab purpose, for cell culture purpose, we use trypan blue because trypan blue helps us differentiate between your live cells and dead cells. So you need to know both. How many live cells are there, how many dead cells are there. So you give your volume based on what the size of your square is. Okay, you should not overflow, you should not give less. You only if you give appropriate volume, you will have your proper number, accurate number. Okay, so this is how your grading on your slide looks. Okay, so this single red box that you see is your 1 mm by 1 mm square. So that one red box can hold 100 nanoliters. Okay, so then you have how many red boxes you can see. So this also you can consider. This red is depicted because those are the boxes we usually count. We do not count all the boxes in this. So you have 3 into 3. So you have total 9 boxes like that. Out of these 9 boxes, we count 5 main boxes. Okay. Then from the number within these 5 main boxes, we use it in a formula to calculate the number of viable cells. And then you can also calculate how many number of cells you need to seed. So every culture dish, you have your 100 mm culture dish, you have your 35 mm culture dish. So all these culture dishes have particular surface area. So based on the area of attachment, you will seed particular number of optimum cells that is required for. Yes, does somebody wants to speak? Okay, so you will, what was I saying? Yeah, so based on the dish, you will seed a particular number of cells in your cell culture plate. Okay, so for that you need to count your cells. So when you count your cells, you can know your optimum number of cells to seed, because if you seed less than, if this, so if you seed, if this is your culture dish, if you, there is x number of cells you have to seed in this. If you seed x or close to x only, your cells will grow efficiently and proliferate. If you seed more than x, then you will have older cells, your proliferation will be slow, your cells will die or grow into senescence, or it will not have that space to divide properly. But at the same time, if you uh, seed less than the number of cells required, then your cells will not proliferate, because your cells need each other to uh, send some signals, secrete some proteins, extracellular matrix. They need to attach to each other, at least a couple of cells, to proliferate and divide and send signals for their healthy growth. Okay, so this number of cells is very important to seed. So that is why you need to always count your cells before seeding. Okay, and how do you count? You count these 5, and that too based on your lab practice or how it is being done in your lab. You might have different ways to count. So here also you should know how to count. Okay, see here, like if it is within your big square, your 1 mm square, nicely without touching any of the edges, then you can easily count them, how many cells are there here. But if it is touching your border, then you should know which side to count, which side to not count. Okay, so what are they saying here? Touching top or right, do count. Touching bottom or left, do not count. Generally this is what we uh, practice. So if it is touching your this top border, like this cell is touching your top border, we will count it that it is within the square. If it is touching your right border, then also we will count it. But when you count these two, you should not count these two. Okay, you should count any of the two. It's not like you should count only this, but you should not count all 4 borders. If the cells are touching all 4 borders, you should not count all 4 borders. First, count the cells within the big square which are not touching your outer boundaries. Then count any 2 boundaries. If you are counting your top and right, you should count top and right to all 5 squares in your hemocytometer. If you're counting the cells that are touching to your left and bottom, then you should count left and bottom only to all your 5 big 1 mm boxes. Okay, so that is how you count. That is your rule for counting cells. So what is your formula now to count? So you have both viable and non-viable cells. Like I told you, you have trypan blue dye, which is helpful in differentiating between your live and dead cells. So your live cells will immediate, how this works is, the cells will be first taken up by the dye will be first taken up by your cells, that it will enter all your cells. Okay, then what happens, your healthy cells will have efflux mechanism. That is, your cells will throw away unnecessary things that is not required. So your healthy or your live cells will throw away your dye, but your dead cells, because it is dead, it cannot function, it cannot throw away anything because it's not alive. So this will still have your trypan blue. So your trypan blue from your healthy cells will be effluxed out, but your dead cells will still have trypan blue. So when you observe under a bright field microscope, you can easily know which cells have the dye, which cells do not have. So based on that, you have to note down how many live cells are there and how many dead cells are there. Okay, because both are important to know your cell viability and then to know how much volume you should seed to get your appropriate optimal amount. Okay, so your viable cell count, live cells per milliliter, that will be your number of live cells counted by number of large corner squares counted. Okay, so that is your this 5 squares counted. So number of cells you have counted divided by your number of boxes counted. Okay, and then you will multiply it with your dilution factor. So when you resuspend your cells, you will dilute in particular amount, and then you will add trypan blue in only that particular amount. You won't add your dye in your entire cell. You will take some 100 microliters from it and then add 100 microliters dye. Okay, in a separate tube. From that tube, you will inject it into the hemocytometer. So that will become your dilution factor. Usually your dilution, you will do 1:1 dilution. So that will become, your factor will become 2. Okay, into 10,000. So this is again based on how much you size it is, how much volume it is, and all that. Okay, and at the same time, your non-viable cells, those are your dead cells. So your dead cells is also similar. Number of dead cells counted by number of large cells in the number of large squares in which you have counted. Same again, multiply it with your dilution factor into 10,000. Then your percentage viability. You will get your percentage viability from this. So your number of viable cells by total number of cells. Your total number of cells is both viable plus non-viable. That will become your total number of cells. So your percentage viability is number of viable cells divided by total number of cells into 100. This will give you your percentage. Usually it will be more than 95%, that is healthy. It will be 98, 99, 99 point so and so. If your percentage viability is very low, that means your cells are not good, your plate is not good, it is either very old or your cells are dying or they have some stress. So it's always better to subculture again from a healthy plate which has a very decent percentage of viability. Okay, now this is how your hemocytometer looks. It is the size of your normal glass slide that you have. At the same time, it will be very thick, and this is your center region. So here only there will be written few things like your depth of it, your size of your square, or the size of your grid, etc., etc. Okay, so here you can see one line here in the center. Here there will be a steel uh, grid or a steel coating on which you will have this grid. Okay, so on this, so here you have these grooves, you can see grooves at both sides. So how you load your uh, trypan blue containing cells into this. So you have a glass cover slip. You will place the glass cover slip on top of it. So it will cover this area. Okay, it will touch this so that your liquid will not move much, your cells will not move much because they are in the suspension. And then you see this groove, you will load your trypan blue containing cells in this groove. So once you load it in this groove, slowly it will go automatically and spread evenly throughout your grid section. Okay, if you pour directly one drop on this and put a cover slip, then it will make a mess. So place your cover slip and load it into this groove, it will spread evenly. Okay, so this is how you count your cells. So you can count uh, different types of cells. Usually your HBC, RBC, you can even look into the microscope and check which one is macrophage, which one is your RBC, lymphocytes, all these. And then you can also at the same time count the number of cells and seed the particular number of cells in your cell culture dish. So the concentration of your cells that you load will be your average viable cells per square into dilution factor into 10 power 4 or 10,000. Okay, then based on that will be your concentration which you have loaded here. Then based on how much you want to load, maybe you got some 10,000 cells. When you calculated you have 10,000 cells in 1 ml or 1 how much ever volume. Then you want to load how much you want to load some 10 power 6 cells in your dish. So you will calculate in 1 ml if you have 10,000 cells in 10 power 6 cells are present in how much ml or how much mol depending on what volume you have taken. Okay, so that is how you count, calculate and seed your number of cells. If you have any doubts, please ask me. Otherwise, we will move on to the next question. Okay. So yeah, as I was saying, different volume, different sizes have different volume. So your 1 mm has your 100 nanoliters, your 0.25 mm has 6.525 nanoliters, 0.25 into 0.2 mm has 5 nanoliters, zero point 0.2 into 0.2 has 4 nanoliters volume. This one single big square. Okay. So let's move on to the next question. Which of these antibodies is used to label most of the neuronal cells? A. NHC antibody, B. MAP2 antibody, C. islet, D. anti-RYR. What is the answer for this?
Map to
MAP2. Very good. So your MAP2 means microtubule associated proteins. So these help in your cytoskeleton. So your MAP2 antibodies which bind to your MAP2 protein. So they are crucial tools for neuroscience for identifying your neurons. These are very important because they are specific to your neurons. So they help in easily uh, identifying your neurons from a mixture of cells, or if you have just dissected out a tissue and you're checking which cell is what, then your map2 is very specific to your neurons, so it is helpful in identifying your neuronal cells. That's why mostly we label the neurons with map2 for checking. Then, since MAP2 is a key protein involved in maintaining the structure, like I said, it is a microtubule associated protein. So, it is very important in maintaining the structure of the neurons, particularly the dendrites. Antibodies against MAP2 help researchers understand how neurons grow, connect, and respond to various conditions. Like I told you, your dendrites will help in signal transmission, and it helps in maintaining that particular shape and the storage of your information in your pyramidal cells especially. So they help in knowing how the cells are branching out like that, or how they are connecting to different cells, how they are transmitting the signals. Based on the signals they get, how they react. So all these kind of research is done using these. So this is your immunofluorescently labeled neurons. So here you can see that in A and D, this is some kind of treatment they have done. This is what you have to see. These are your antibodies that are used to label MAP2 and picophilin. Okay, so both are your cytoskeletal associated proteins. Your picophilin is your actin polymerization protein. So your MAP2, you can see your entire neuron is stained with your MAP2. You can see the axon, the dendrites, everywhere wherever the cell has to maintain the shape, it has MAP2 protein. So that is why you label them with MAP2. And this is how they look. And when they co-labeled it with MAP2 and picophilin, you can see that both of them are labeled in the cytoskeleton, and how different cells have different kind of localization for this. Okay, so next is, which of these do not which of these is not a developmental stage in rats? A, embryonic; B, prenatal; C, rental; D, neonatal. What is the answer for this? Yes.
Option C.
Option C, yes. So this is, I think, pretty easy. So your rental is not any of the stage of development in your rats or any other. So your embryonic, prenatal, neonatal are your different stages. Your embryonic is since when the embryo has formed, and then your prenatal is at your stage before the birth, and then your neonatal is your stage immediately after the birth. Okay, so the answer for this is rental, which is not a developmental stage in rats. So rats, why rats? Because rats are very useful as a research model. Okay, rats, mice. So and rats are more similar genetically with humans, and rats are widely used in scientific research because their development is relatively quick, and they share many biological and behavioral similarities with humans. So not only genetic or molecular level, even their behavior is also very similar to humans. That's why rats are favorite when it comes to animal models. Each stage of rat development offers valuable insights into fields like neurobiology, behavioral sciences, genetics, pharmacology, cell biology, clinical studies, etc. Then understanding your phase trials also are also done in rats. Then understanding these stages is important for researchers when designing experiments that study age related factors such as learning, memory, responses to drugs, etc. So why do you have to know the different developmental stage of rats? One, you can be studying the developmental stages itself to see at what stage what is happening. But even if you're not doing developmental studies, you should know the stages, because whatever experiment you're doing, or whatever proteins or signaling or condition you are trying to work on, you should know at which stage you have to work on. Like for example, if you're doing growth related studies, if you're doing studies that is related to children or something, then you should know at which stage you have to take the cells and uh, isolate the cells from the rat to get similar kind of tissues or similar kind of proteins. You cannot take a adult completely grown rat and then correlate it with children's study to give you a result. That will be wrong. So if you want to know adolescent stage of human beings, if you're studying something at puberty level of humans, then you should also take a adolescence stage of the rats. At the same time, if you're studying age related conditions, if you're studying age related diseases like Parkinson's, Alzheimer's, etc., then you should also take adult aged rats. So you should know which stage you have to do according to your experimental design. That is why it's important to know what is the uh, mating time of rats, what is the, if you're doing the knockdown kind of studies. If you're growing transgenic mice or anything, you should also know what is the mating time. You should also know, what is the gestation period. You should know what is the number of litter size, healthy litter size. You should know the time for birth. You should know which stage what is going to happen. Okay, so this is your simple chart to know your postnatal stages. So before your postnatal will be your prenatal, and then before that you'll have your embryonic. So postnatal, 0 is the day of birth. From there will start your postnatal. First will be your neonatal from 17 to 14, and then your weanling stage, then your peri-adolescent stage, and then your young adult stage. Then later it will go into adult stage and then aged. Okay, so your P7, P21, these are all denotions that they give. Okay, and then you have to also monitor the weight of your rats. You have to monitor if they are developing any diseases, to avoid environmental factors affecting your study. So this is how they look in real, different developmental stages. So this is all your postnatal stages like I showed you here. So this is your neonatal, this is your weanling stage, this is your peri-adolescent stage, this is your adolescent stage, this is your adult stage. Okay, so this is how they look. So at which stage what you have to collect, what kind of treatment you have to give, you have to know. And like how they have taken images there, if you're doing studies on developmental stages, you have to monitor their growth at different stages. You have to monitor their weight, size, if they're growing healthy or not. Okay, next question is, which of these protein misfolding and aggregation is linked to Alzheimer's disease? A, amyloid beta, B, tau, C, both A and B, D, none of the above. What is the answer?
I'm option C, both A and B.
Option C, both A and B. Okay, so that is your correct answer. Very good. So your amyloid proteins are which which have this fibril like structure, and they aggregate into insoluble fibers. Okay, so they are associated with this Alzheimer's disease. At the same time, this tau protein is also associated with the Alzheimer's disease. So this is your some mechanistical things when they do Alzheimer's disease, how this tau protein plays a role. Okay, tau phosphorylates, dephosphorylates, and then hypophosphorylates. For different types, uh, there is again different effects. So you have again your tau kinases, then you have your tau phosphatases. Kinase is the one which phosphorylates. Phosphatase is the one which dephosphorylates. So based on the phosphorylation on your tau protein, again different effects are seen. Okay, and then even these also have this fibrillar structure and all this. Okay, so then not only Alzheimer's, tau related, there are other diseases also, neuronal disease. They are age related and they are called as primary age related tauopathy. Okay, so they affect different regions of your neurons. Different protein aggregations are formed. They are misfolded. Your protein has to be properly folded for it to function, especially when it has to have binding to other proteins, or it is, if it's enzyme, then definitely the activity is gone if it's not folded properly. But even if they are adapted proteins, they have to participate in the signaling. And then if they are structural proteins, they have to attach to other proteins to maintain the structure. So everywhere the folding of the protein plays a major role, and these two proteins are very important in this protein misfolding and aggregation. If there is a problem in this, or there is some phosphorylation defects or anything, then your proteins will get misfolded and aggregate, and then they will be useless. They will not attach, they will not function, they will not have any activity, and that So one protein is not having just one role. It will be a part of several signaling pathways or several structural features. So if it is a point for many different other things that will be spreading, branching from it. So if one protein also is having a problem, you will have obstacles, or you will have pause in many different pathways within your cell. So protein, and when such proteins which help in folding of other proteins, if they are itself mutated or having an issue, then many other proteins will uh, be non-functional, and then the issue will escalate exponentially. Okay, some of your amyloid proteins are like your prion proteins, your synuclein, you might have known, or your amyloid beta peptides, etc. Okay, so both tau and amyloid are important. Next, which of these brain regions is associated with memory and learning? A. Hypothalamus, B. Amygdala, C. Visual cortex, and D. Hippocampus. What is the answer?
hippocampus.
Hippocampus, very good. So I think only one person is answering everything. I want every one of you to try at least. If not, okay, I have not forced. So your hippocampus is your answer. We saw it in the first question itself. It is involved in learning and memory. So hippocampus is essential for new memories, learning, cognition, navigating spatial movements. So not only memory and learning. So learning, everything involves learning. Everything, learning covers everything. So particularly if you see cognitive abilities, your navigation, your spatial environments, just be aware of the spatial zones of your surroundings. All these are related to your hippocampus. Damage or dysfunction in the hippocampus can lead to memory loss, obviously, excuse me, learning difficulties, and conditions like Alzheimer's, as a highly plastic part of your brain. So here, plastic part of your brain means it's a very dynamic part. It is also a key area of interest in studies of brain health, neurogenesis, and cognitive function. So it's very important. Hippocampus is especially in your cognitive research, hippocampus is very crucial. So this is your brain, and this is where the hippocampus is. Then you have your thalamus, you have your amygdala, you have your cingulate gyrus. Okay, so mainly we should remember hippocampus, short term, long term memory, learning, spatial memory. Okay, and then little interesting facts about your hippocampus, because we got two questions on hippocampus. The name, why is it called hippocampus? It is derived from Greek word hippocampus with a K. So hippos in Greek, not English. In Greek, hippos means horse and campos means sea monster. So you can see the shape, maybe not in this cartoon, but in your first question, I showed an image. So yeah, so here you can see the shape of your hippocampus, how it is. So this looks like a seahorse. So based on the visual appearance of your hippocampus, they have named it a seahorse. Hippos means horse, campos means sea monster. So this is how your seahorse looks, and this is how your hippocampus looks. Two hippocampi are located just above each ear, one in each hemisphere of the brain. Part of the brain system responsible for regulating emotional responses. So along with your memory and learning and all this, it's also important for your emotional responses. Anything, your happiness, sadness, stress, anxiety, everything is related to your hippocampus. That's why all your recent anxiety related, depression related research, and emotions and all this are based on your hippocampus, involved in processing memory and storing them, full of stress receptors and holds on to fear memories of the past, playing a role in PTSD and phobias. So, as it is involved in emotional responses, fear is also one of your emotions. So, your fear, your pain, your phobia. So, because it has many of the stress receptors, your anxiety, your depression, whatever is there, everything is being processed by your hippocampus. And it also, why do you have phobia? Why do you fear something that you have already seen or maybe even not seen sometimes? Because it stores your fear memory also. When you see something, if you're scared of a lizard, your brain, hippocampus stores that you're scared of lizard. So again, next time when you see a lizard, you're scared again. So that is all done because of hippocampus. It's not just happening, it's a mechanism because it stores your fears. Okay, and that is how your PTSD is developed, your phobias are developed. Your PTSD is post traumatic stress disorder. So after a major trauma, like something like very uh, traumatic incidents, like a huge accident, or loss of someone, uh, moving to completely different place, or people who were in the armed forces, once they come back from their regiment, because they have seen so much during their time period, people who are you know suffering from some kind of violence. So all these things also is stored in as trauma in your hippocampus. So then this will even if you come out of that traumatic situation, you will still have the residual stress of that trauma in your brain. So you're still not getting that trauma, but your brain will always have some kind of triggers, some kind of responses, some fears, some imaginations, hallucinations, kind of thing. These are called your post traumatic stress disorder. Okay, all this is also hippocampus related. Then it is one of the first parts of the brain to suffer damage in memory related conditions like Alzheimer's. So, Alzheimer's, your Parkinson's, all this memory related diseases, your hippocampus is key because it is what stores memory. So, let's see what the other things are. Your first option is your hypothalamus. So, your hypothalamus is your hormone secretor. So it is where it is the regulator for major hormone secretions, and it also maintains your body temperature, then your hunger, your thirst, your fullness after eating. Everything is because of hormones that is released by your hypothalamus. And then your sleep cycle, your mood swings, all these are also hormone related, linked to your hypothalamus. And it also regulates your heart rate, your blood pressure, your body temperature, etc. So basic your body homeostasis. Okay, so the next one is amygdala. What does amygdala do? Amygdala also, like, here we saw this emotions. So your amygdala is also plays a key role in maintaining your emotions. Okay, and whatever response you give to emotions are also because of your amygdala and how it processes your emotions. And then it is also responsible for your flight or fight or flight response. Like if there is a situation, you're scared or it's a dangerous situation, do you fight or do you flight? Flight is run away. Do you stay and fight or do you run away from that situation? This decision is also made by your amygdala. So you will know that few people, when there is a tough situation, they run away. They, I don't want to do this. And then few people are like, let's face it. So that is your fight or flight response. And then your decision making, your cognition also comes under your amygdala. Okay, so major feature of your amygdala is your fight or flight response, because that is Also sometimes individual to a person. Few people have more fight trigger and few people have more flight trigger. Next is your visual cortex. So visual cortex is, as the name suggests, it is where your visual information is processed. Okay, so your it is present in your occipital lobe, and it processes whatever features you see through your eyes, like some shapes or colors, or some the spatial areas, etc. Okay, it is to and not only just receive it, it will also perceive that information. It will integrate your visual information whatever it gets, and then it will send to other parts of your brain to analyze what it is, especially in this analytical kind of situations. Okay, and basically, so it also helps you in recognizing something that you have seen again. So that is your visual cortex. Your next question is, neurobasal medium is buffered by what? Basic amino acid, B oxygen, C sodium bicarbonate, D glucose. What is the answer for this?
Sodium bicarbonate.
Sodium bicarbonate. Very good. So, what is a neurobasal medium? A neurobasal medium is a neuronal cell culture medium that is designed to support long-term cultures of postnatal and adult primary neurons without the need for an astrocyte feeder layer. In simple terms, sodium bicarbonate in neurobasal medium acts like a safety net for your pH. So basically, it's your pH buffer. So usually we use the CO2 incubators like we studied before. So this is like an alternative for your CO2 incubators. If you don't have a CO2 incubator, you will use your sodium bicarbonate. Uh, but it's not foolproof. It will not buffer it for a long time. It eventually needs CO2, but for the time being, you can manage. It ensures the environment remains suitable for neurons to thrive by preventing the medium from becoming too acidic or too basic. This stability is crucial because neurons are delicate and need specific conditions to grow properly in the lab. So, your neurons conduct your electrical impulses. So, if your pH of your medium in which they are growing, if it's if there is a slight change in it also, your neurons will not grow properly. And if you're checking some impulse studies or some signaling studies, then it will have variations in your results. So basically, it's for your postnatal and adult primary neurons that you isolate, and then you have to also maintain your this glucose concentration, your osmolarity, everything. Okay, so one other thing that was here, astrocyte feeder layer. So for astrocyte feeder layer is it supports your differentiation of your neuronal networks generally. So when people want to grow neuronal cells, they also give a astrocyte feeder layer which helps your neurons to network properly. Okay, so your feeder layers also can come from your primary cells of astroglia, or it can be cryopreserved and then be used. So but that is one extra cell in your experiment now. So if you don't want the involvement of your astroglial cells, then you have to maintain your this neurobasal medium in your particular buffered conditions and give it all the necessary things that are required for it to network and grow and signal properly. So this is your basic media, how generally all your mammalian media looks. Your neurobasal media also looks like that. And these are your components of your neurobasal media. It has all your amino acids, arginine, aspargine, cysteine, isoleucine, leucine, phenylalanine, threonine, etc. And then it also has your non-essential amino acid L-nitroglycerin, and other major important components are your D-glucose, your hepases, your phenol red. Phenol red is your indicator for change in your pH. So how will you know if even if sodium carbonate is there, whether for how long it is maintaining your pH and when it has changed? So you need to have your phenol red indicator to know your change in the pH. Then you have your sodium pyruvate, then other trace elements and vitamins. You have your organic salts, which are very important for your cell function, because your Mg plus 2, Ca plus 2, all these are cofactors for different proteins and enzymes. So your inorganic salts are also very important, and your zinc, your ferric ions, etc. for your even other processes like your endocytosis, exocytosis. All these are involved. Zinc and ferric and calcium are all involved in this, and your sodium chloride and your potassium chloride to maintain the membrane potential, etc., etc. If you want to know why each and everything is used, you can visit this website, or you can visit any like normal website which provides cell culture media and which provides this neurobasal media. Within the catalog of that media bottle, you will have what are the components, and then you can see what is used for what. Okay, so anybody has any doubt, you can please ask me. Okay. No. Okay, so next question. Read the following statements carefully and then answer. Xeno-free medium. A, it does not contain ingredients derived from non-human animals. B, does not contain recombinant materials made from non-human animal DNA sequences. C, may contain purified, processed, or unprocessed material from human sources. D, may contain recombinant material from human, plant, bacterial, or yeast DNA. E, animal derived components may have been used as raw materials at the secondary or tertiary level of manufacturing unless otherwise indicated. So read it properly, and then your options are: A is false, uh, next is D is only true, next is A, B, C are true, next is all A to B, that's your A, B, C, D, all are true. What is the answer for this?
Ma'am, is it C?
C, may contain purified, processed, or unprocessed materials from human sources. Okay. Does anybody have any other answer? So, do you know what is a xeno-free media first? What does xeno mean? What does xeno-free media mean? Tell me. Do you know what xeno means? What does the term xeno mean? Somebody just answered C, right? So you must be knowing, like you might, or you might have an idea what is xeno-free medium or what is xeno. Do you know? Yeah. Does anybody know what xeno is? Xenobiotics. We know, right? We use this term. Okay, fine, no problem. So, xeno means something that is foreign, or something that is not related to you, or especially here in biology, it means something that is of a different species. Okay, so xenobiotics means something that you do within the same species. So here your xeno-free media means your whatever materials you use to grow your mainly for human cells or human tissues, everything should be from your human source. Okay, mostly mainly all the primary raw materials that you use, that should be from your human source. But your tertiary or secondary level sources can be from your animal derived, but your DNA sequences should not be from your animal sources. So it can be plant yeast source for DNA. The raw materials can be processed first properly so that it does not have any species contamination, and it can be processed to a secondary tertiary level, and then it can be used. So in that way your xeno-free media is made. So the answer for this is D. A to E all are true, so you have to read this very carefully. Everything in this is true. So your xeno-free media does not contain ingredients derived from non-human animals. So your main ingredients like your serum, your albumin, or your vitamins or whatever main ingredient that you add in it should not be directly taken from non-human animals. Second, it does not contain recombinant materials made from non-human animal DNA sequences. So you can use recombinant materials in this, but the source from which you have made the material, like you need a DNA sequence, you need a gene sequence to make some recombinant material, like your recombinant hormones or your recombinant, like your recombinant insulin, something. But the sequence from which it is made should be your human sequence. It should not be some other animal sequence. Next is may contain purified, processed, or unprocessed material from human sources. So obviously it's for human cell growth. So you can take anything from your human sources. Your it can be purified, you can be processed, and it can be unprocessed also. But because it's from the same species, you can always use it. Next is may contain recombinant materials made from human, plant, bacterial, or yeast DNA sequence. So here you should see that it is not made from non-human animals, but it can be made from plant, bacteria, yeast DNA. Okay, so you can make materials from there because they're completely different class and different genus as well, but you cannot make from a different animal. Okay, because your mass production of your recombinant things will be usually done in your bacterial or yeast system, so that will not affect because your genes in your bacteria, yeast, plant are all this prokaryotic genes, so they will have different type of genetic makeup and arrangement. So that is fine, but your animal sequences are very similar to human sequences, so that you cannot use. Next is animal derived components may have been used as raw materials at the secondary tertiary level of manufacturing unless otherwise indicated. So yeah, so in few cases you can use the materials which are processed from the animals. Like if you want to take some material that from an animal source, it cannot be your primary raw material. It cannot be something that you just isolate, purify, and add. No, it should be processed to certain levels with certain conditions where that specificity to that species will not be still present in it, so that it does not generate any kind of antigenic response from your human cells. So it should be processed to 2 3 levels, so your secondary tertiary levels, and then after that final processing you can add it only if it is needed for your experiment. If it's not if it's not required, you can also have a media which does not have even at secondary tertiary level manufactured animal derived components. So this is all true for your xeno-free media. Xeno-free media is a type of cell culture media that is free of components from strange species or your foreign species. or xenocomponents. So xeno means foreign. In cell culture, human cell lines that are cultured using human derived components like human serum. We usually add serum in the media. It's your blood serum component, okay, and are considered xeno free because it's from humans, it's derived from humans. But in our normal cell culture, we do not use xeno free for everything. Normally, other cultures we usually use FBS. FBS is a fetal bovine serum, okay. So, and then we also have some other kind of components which are also animal derived. So in those cases, if your experiment is not does not have a requirement of xeno-free media, you can use it. But if it's a xeno-free media, even your serum, your other components should be derived from a human source and are considered xeno-free because there is no difference between the species. Xeno-free media are used in cell culture applications to avoid the risks associated with animal-derived components and pathogens found in fetal bovine serum. Like I said, we use generally fetal bovine for normal cell culture. Some, even though they are like purified and they are sterilized and they're distilled and all, sometimes maybe there might be some pathogen that might pass all these checkpoints and still be present in your fetal bovine serum. So that is the reason we when you are very crucially working on xenobiotics or xeno research, you need to use xeno-free media wherein you will use serum from human source itself. But because the source is human, it's very uh, rare for production, or it's very um, technical and tricky. So xeno-free media are used for a variety of cell types, including human-induced pluripotent stem cells, mesenchymal stem cells, and T cells. So you can see the type of cells for which xeno-free media is a requirement, because you're making induced pluripotent stem cells. So you will make these cells so that they will differentiate into something else. They will differentiate it to some other injured cells of your body and it will replace them. So when they are being made a part of your body, then you cannot uh, use something that is outsourced other than human species. Same for mesenchymal stem cells and T cells, especially when you're doing some antigen response kind of things, or you're developing some antibodies or something that is very specific and crucial to humans, then you cannot take the risk of creating an antigen from animal components. Okay, so this is very important for your xeno-free medium. Okay, if anybody has any doubt, you can ask me. Okay, if there is no doubt, we'll go for the last question. So, Leibovitz L15 medium contains dash but lacks dash. What is the answer for this? Are you guys there? What is the answer for Leibovitz L15 medium? It contains what and does not contain what? Your options are sodium. It contains sodium pyruvate but does not contain sodium carbonate. It contains sodium tartarate but does not contain sodium bicarbonate. It contains sodium glutamate but does not contain sodium bicarbonate. It contains sodium pyruvate but does not contain sodium bicarbonate. I think A and B, D are same option. So you got like a 50-50 here. One option is gone. You have to choose between only three. What is the answer? Anyone still there?
Might be sodium pyruvate and sodium bicarbonate.
Sodium pyruvate and sodium bicarbonate. Okay, let's see if you are right. So it is sodium pyruvate and sodium bicarbonate. So what is your Leibovitz L15 medium? A Leibovitz L, it's a kind of medium that you use for your primary cells and monkey cell lines. So it contains usually 2 L-glutamine, your amino acid, and no sodium bicarbonate. This formulation is designed to be used without additional CO2 in the atmosphere of the cell culture. It has applications for cell culture, cell growth, viability, and 2 glutamine and no sodium bicarbonate. So your sodium bicarbonate, uh, it helps in buffering, and it maintains for a particular time, and then it needs your CO2 for your adjustment of adjustment of your buffer. Where you don't want to cultivate it in CO2 at all, then you don't need your sodium bicarbonate. So your L15 medium supports growth of HEP2 monkey kidney cell lines and your primary explants of embryonic and adult human tissue. Embryonic and adult human tissue explants, like that is tissue culture. Take a part of your primary tissue and then you grow it. Okay, so this also looks similar to your normal mammalian culture. Okay, so that is all for today. Does anybody have any questions? You can ask me any of the answer if you think you have some doubt, or anywhere you want me to explain again. Anyone? Do you have any doubts? Do you have any questions that you want to ask? We still have some time left. Okay, then let's just revise all the answers. So, what is the characteristic shape of hippocampal neurons? It is your pyramidal shape. They have the particular structure for learning and to maximize the efficiency of signal processing. They help in learning and memory. Key points. Next is papain is a dash derived from dash. So papain is a proteolytic enzyme, proteolytic protein lysis. Remember. Then it is papain, so papaya, you can easily remember. It cleaves your peptide bonds in your proteins. It is used in digestion of your extracellular matrix, so it's helpful in isolation of your cells without damaging your cells. Next is your hemocytometer. This is very helpful and handy and useful in your when you're culturing the cells in practical. So you should remember this always, and this formula should be by heart, and you should know to calculate. The quick calculation should happen. So in hemocytometer, the volume of 1 mm is to 1 mm square is how much? It is 100 nanoliters. So this is how your hemocytometer looks. This is a thick glass. You have a grid here in the center. You place a glass cover slip. You load the cells containing tryptophan blue, which will help you in differentiating live cells and dead cells. And then uh, once you load it in this, you will start counting the cells in the 4 corner big squares and 1 center square. So while counting also, you should remember, first count all the cells that are inside your big square, and then you should count any 2 borders, either top right or bottom and left. The cells which are touching to these borders. Do not count the cells that are touching to all 4 sides. And if you are counting top and right border touching cells, then count top and right for all the 5 squares. If you're counting bottom and left, count bottom and left to all the squares. Okay, that is the technique. And how your trypan blue works: it is a dye your enters your cells. Healthy cells have an efflux mechanism. It will flux your trypan blue outside your cell because it's foreign to your cell. It does not need it. And your dead cells, because they are dead, they cannot efflux it, so they will still have your trypan blue. That's how you can differentiate between your dead and uh, healthy cells. So a viable cell and non-viable cell count is important. You have to calculate it per milliliter, and the formula for that is number of live cells by number of large squares counted. It's usually 5 large squares, and your dilution factor usually 2, because you will use, like, if you take 100 microliter of your cell suspension, you will take 100 microliter of your trypan blue, and you'll multiply it with 10,000. And then same for non-viable, number of dead cells by number of large squares counted, and your dilution factor into 10 power 4. And then your percentage viability will be your number of viable cells by total number of cells counted. Your total number of cells is viable plus non-viable into 100. So this is your percentage viability. It should be higher than 95%. It should be 98, 99. Then your cells are very healthy. If it's very less, that means you have more dead cells than live cells. Then it's not a good uh, set of cells. Okay. So always count your cells before seeding. Next is which of these antibodies is used to label neuronal cells. So your MAP2, you'll answer. Your other options are your MHC. MHC, you know, it's your MHC class class proteins. It is present usually in immune cells. MHC, HLA. So they help in antigen presentation, identifying the type of antigen, and then class 1, class 2, all this comes into picture. So it's basically for your immune system. MAP2 is for your neurons. They are microtubule associated proteins, and they are present in your neurons specifically, so they help in identifying neurons. C is your islets. Islets are your cells from your pancreas, so any antibodies for your pancreas is your islet antibody. And then anti RYR. RYR is your um, rhinodine receptors that are present in your endoplasmic reticulum of all your cells. So these endoplasmic reticulum stores calcium. It's a calcium store. So usually for your muscular cells, or cells that need contraction, relaxation kind of processes, your cardiac cells and your muscle cells, the RYR is very important, because your calcium is involved in your contraction and relaxation of your cells, and RYR only supplies that splurge of calcium from your endoplasmic reticulum when required, okay, from the storage. So your MAP is a microtubule associated protein. It is involved in maintaining the structure. It helps in your dendrite branching, connection, and growth. Okay. Next is your, which of these is not a developmental stage of rat? The answer is not rental. So embryonic is your developmental stage, prenatal is your developmental stage, neonatal is also your developmental stage. So you should know different stages of development, because according to the design of your experiment and need of your hypothesis, you should know which stage of development you have to choose and proceed with your experiment. Okay, so this is how your rats look at different stages of development. First is your neonatal stage, then your weanling stage, then your peri-adolescent, adolescent, adult, young adult, etc. Okay. Next, they are a very good model for different behavioral, neurobiological, genetical, molecular, pharmacological, clinical studies. Next is your, which of these misfolding and aggregation is linked to Alzheimer's? So the options are amyloid beta and tau protein. So both your amyloid beta and tau protein are involved in Alzheimer's disease, because both of them are important for protein misfolding and protein aggregation. So when you have misfolded proteins, they generally aggregate because they lose their function. So if they are signaling proteins, their signaling pathways are affected. If they are structural proteins, the structural integrity of the cell or the region will be lost. If they are enzymatic proteins, then the activity of the protein will be lost. And in all these cases, it affects the cell in many different ways. So the tau related are very important. They are also present in age related diseases which are called as taupathy. So different neuronal diseases are also associated with this. Here next is again your hippocampus. Which of these brain regions is associated with memory and learning? It's your hippocampus, and your hippocampus name is from Greek, meaning horse of sea, like seahorse. And then because it looks like a seahorse, and then it's responsible for your regulation of emotional responses. It is involved in processing memory and storing them. It is involved in emotional responses, your PTSD, anxiety, phobias, etc. It is one of the major parts to suffer in most of the memory related brain diseases. Okay. And other than that, hypothalamus we spoke about. It's the regulation of your hormone secretion. Different hormones are secreted for different purposes: your sleep, your hunger, your thirst, your mood swings, etc. So all these hormones are in under the regulation of hypothalamus. Amygdala is your flight or fight response. It is also related to your emotional responses, your anxiety, etc., your mood. Then your visual cortex, it processes, it receives information, visual information, it processes it, it sends signals to store it, and it helps in recognition, etc., etc. Okay. Next is your neurobasal medium. So here we spoke it's for the neuronal cell culture, for postnatal and adult primary neurons, and uh, it is buffered using sodium bicarbonate, which helps in maintenance, and then you have your CO2 for your sodium bicarbonate to buffer. And then stability of your neurons is very important because the buffer in which they are there, depending on that, their responses and their growth and their metabolic functions will change. So these are the components of your basic neurobasal medium. Essential amino acid, non-essential amino acids are the components, vitamins, trace elements, your inorganic salts, etc. Next is read the following statements carefully. Sorry. Uh, Xeno-free media. So, what is your xeno-free media? It is a media where you culture your cells free of strange or foreign species, materials, or components. So, all the basic raw materials are human derived, including your serum, and you cannot take risk of contaminating it with pathogens or any other even animal derived antigen or components in this. And it is mostly for your stem cells and your immune cells because you want them specific to humans species. So the main components of this is it should not contain ingredients derived from non-human animals. It should not contain recombinant materials also made from non-human animal DNA sequences. May contain purified, processed, unprocessed material from human sources. May contain recombinant materials made from human, plant, bacterial, or yeast DNA sequences, but not animal DNA sequences. Animal derived components may have been used as raw materials at the secondary or tertiary level of manufacturing, which are processed and or unless otherwise indicated, mostly that also not be used. So your answer for this is all A to E are true. Uh, your last question was your Leibovitz L15 medium, what it contains and what it lacks. So it contains sodium pyruvate and it lacks sodium bicarbonate. So option D. So L15 medium you use for the Hep2 monkey kidney cell lines mostly, and your primary explants from your embryonic or adult human tissues. You generally do not grow this in a CO2 incubator, so you do not add sodium bicarbonate. But it majorly contains L-glutamine and sodium pyruvate. Okay, so this is how it looks. So this is your last question. If you have any answers still, you can ask me. I hope it was understandable and helpful for you all. Okay, so if you guys do not have any questions or any doubts, you can exit the session.
Thank you, ma'am.
Okay, thank you, everybody.